Hello students, at this time where we are actually trying to come out of the grips of coronavirus as the whole world is suffering from coronavirus issues at the moment. So in this particular video I would like to focus on talking about in relevance with the coronavirus infection. Okay, what type of questions are possible in the exam? So keeping in mind that for both APPSC as well as for the TSPSC future exams, it is in the syllabus that we have both health and disease as well as vaccine development diagnostics are part of it. So in connection to that, there are many developments that are happening and many related things are coming in the news related to coronavirus. Okay, so my goal in this video is to walk you through some of those aspects and explain you the principles behind them so that you will get benefited from understanding this information and practice writing some answers with respect to that. First of all, we have been hearing that coronavirus is a type of infectious agent which affects the respiratory system of human body. If you see the description wise, coronavirus looks like an enveloped virus and it has spike-like structures on them. Okay, so these spikes, what gives rise to the appearance of a crown, what seems to resulted in the term corona and the name coronavirus. It is an RNA virus, meaning it contains mainly RNA as a genetic material, and they sometimes say that it has positive sense strand RNA, meaning that this RNA acts like a sense RNA and as an mRNA, thereby initiating the process of protein synthesis from it. Okay, but of course, coronaviruses are not new viruses. In the past, we have a SARS coronavirus, which resulted in an outbreak in China in 2003, and similarly MERS, Middle East Respiratory Syndrome coronavirus. And we have seen an outbreak of this in Saudi Arabia in 2012 and also an ongoing problem in some regions. And the latest one being the 2019 coronavirus infectious disease or COVID that we refer to as, which had begun in China in 2019 and shaking the whole world. Okay, so they can ask questions on overall description of the coronavirus, like structure. They can ask questions with respect to the what type of symptoms it can cause, origins of it, because we know that there is, of course, not a clear evidences with respect to it, but it could be a zoonotic disease, meaning it probably had spread from animals to humans. And, probably they were also suggesting that it could be coming from as bats as hosts or pangolins, but these things need to be further verified. And we know that symptoms wise, it affects respiratory system. There are, some cells in the respiratory system which have receptors, okay, to which coronavirus binds, causing common cold-like symptoms and also pneumonia-like symptoms, where fluid gets filled in the alveolar regions of the lungs. Okay, but the studies are suggesting that it is also has ability to infect various tissues and various parts of the body as well. Okay, so questions can come on these aspects of things, and as well as we have been hearing quite a bit about diagnosis. We know that ICM are recommended to use what is called reverse transcription based PCR for diagnosing corona infection. So in PCR, you might have heard about that because we study this under immunodiagnostics in Unit 7, where this is a molecular diagnostic tool. So PCR stands for polymerase chain reaction. So it is a DNA amplification tool. So here in this machine acts like a Xerox machine of DNA. If you put the DNA into that, it will make multiple copies of DNA and providing genetic fingerprint as a pattern of information specific to this sequence of a pathogen. Okay, so what do they do? Let's have a brief look at this. So for the sample, they take a swab, that is a cotton ball, and a sterile cotton ball they put to the throat, okay, or the nose, collecting some sample containing the virus in it, from which they can extract RNA, and this RNA has to be converted first into DNA. This is where the concept of reverse transcription comes into the picture. Because in our bodies, we have DNA as a main genetic material and we create RNA from that in a process called transcription. As this is reversed to that process, it is called reverse transcription. And then what do? This DNA is used and placed in the PCR and we mimic the process of DNA replication by adding all the required components to perform replication process. And most importantly, primers. So what are primers? Primers are complementary sequences or nucleotides to this DNA sequence, meaning if this contains A, primer will contain T. If this contains G, primer will contain C. So like that, a stretch of nucleotides which bind to this DNA are taken. Only when primers bind to this sequence, DNA replication process will begin, and that eventually resulting in the amplification of DNA, which is a sign of a positive test. If no such amplification takes place, meaning that this person probably don't have that virus in his sample. Okay, so this is a basic principle here. So as the primers have to bind and multiply, so only when there is a match with respect to that, it will result in the multiplication. So this forms a basis for RT-PCR test for coronavirus. And also they can ask you questions on critically discuss the advantages and issues with respect to such type of diagnostic tests. I'm highlighting few points here. So this is a highly accurate test because as I was mentioning to you, the primers have to match the viral genetic material. Otherwise, there won't be any amplification. So this is really accurate. Next, sensitive. So even small amounts of viruses present in the patient sample, they are sufficient to result in this positive test. So it is easy to identify. So it is very sensitive. But the problem with these type of tests is it is expensive. They are saying that around 4,500 or so it costs. So several thousand rupees here, and also time taking. Understand the things here that you have to extract the RNA, convert into DNA, and then amplify it, which will take an hour or so. And all together, keeping in mind the logistics here, that where testing have to be done, analysis has to take place, the samples have to be sent to the laboratories, which will take close to a day or more, right? So in that sense, it is not very quick test and time-taking test. Also, we are hearing about rapid antibody diagnostic tests. Kits. ICMR, Indian Council of Medical Research, has initially suggested to use rapid antibody diagnostic kits, of course, because of a problem from some of these companies who sold and we imported those tests from them. So there was some technical issues with them. Okay, otherwise, what is a principle, basic working principle behind this? In these type of tests, what we are detecting is antibodies. So they take the patient's blood sample, and inside the blood sample, specifically in plasma of the blood sample, where antibodies are present, they will look for their presence. And probably you have studied this in immunity component in Unit 8, that in our immune system will respond to antigens and infectious agents by producing antibodies. This is one method of protecting our bodies. So here we are detecting those antibodies which are produced in response to infectious agent. So I've taken an image of a company's kits. How do they work here? So what do you notice here? They load the sample from the patient. The sample will migrate to these areas. You see here 2 points, test and control. In the test, what normally happens is the sample they will load contains antigen. So here in the kit they will add antigen specific to coronavirus. So sample should contain antibodies. When antibodies react with the antigen or bind to the antigen, it develops a signal like this. So if you see a signal along with the control, because control is used to make sure that this test kit is working as expected. Okay, so when you see these two bands, it is positive test. If there is no such band for the test portion, it is negative. But if there is no band for the control, then this is failed. The kit has not worked. Okay, so this is how they perform the antibody diagnostic kit, as it is mentioned to be a rapid test. It would only cost us few minutes, about 15 to 20 minutes, or sometimes few minutes more, but in the range of minutes. And it is also less expensive. It won't cost much, I think 10-fold less compared to the RT-PCR approximately. And also this is a very good tool for monitoring hotspots, because hotspots may contain several infections spreading quickly, and these type of rapid tests are suitable for that. And additionally, as this can also show us the asymptomatic patients, meaning those people who don't show symptoms, even though they are going through coronavirus infection. In those cases, what normally happens here is they probably still have immune response working, so we can still be able to detect the antibodies in them. So this is a very useful benefit of that. But the problem here that you cannot and may not identify active infection. You know why? Because a person who is going through the infection, he might have not developed antibodies. So if you do the test on them using rapid antibody diagnostic kit, it will not show the positive test for that. Okay, so it is not suitable for that. And also on top of this, if someone shows the positive test for this, we have to recheck this and reconfirm this using RT-PCR test. So this is the basic idea about rapid antibody diagnostic kits. Know it because sometimes they can ask short notes on this, but these are the key things they can ask you related to diagnostic kits in the exam. Next, the next thing is, as part of immunity and vaccines, we hear that for controlling corona, many scientists around the world are actively working in developing vaccines. And vaccine is a preventive measure. So before getting the infection, we have to take vaccine. So body gets prepared towards that, thereby preventing the problem. So it is a very, very important, and we're hopeful that these companies would become successful, but we don't know whether it's going to take months or years. But given that many companies are working, maybe soon we'll hear a good news on this. So what categories of vaccines? And again, in immunity vaccines, you might have studied about different categories of vaccines. We have traditional vaccines as per the WHO list of vaccines that are under development for corona. There are live attenuated vaccines, there are inactivated vaccines that are in preparation, and also modern vaccines like subunit vaccines, where portions of the vaccines are used, or vector vaccines, where you actually take vector and genetically modify so that it doesn't multiply, but it presents the antigen to our immune system. Similarly, they are considering using DNA and RNA components of the virus to act like vaccine candidates here. Okay, so also we have been hearing about multiple attempts and works that are taking place with respect to the treatments. But so far, we are not talking about specific drugs being developed for corona. What most of the drugs that we're talking about here are already existing drugs and meant for treating other infections, like antiviral drugs. They're talking specifically about anti-HIV drugs, antiretroviral drugs. Okay, these drugs, because of some similarities in the virus, may act. We are hopeful to that. Also, like ICMR had initially approved some of these things, but they are not working really well. And the anti-malarial drugs like hydroxychloroquine is also considered to be potential, and ICMR suggested to use them on high risk group of people. They are suffering severely from this, but it is just a potential, a possibility of this. And also some organizations are working on anti-malarial drug with antibiotic called azithromycin. Seems to be little bit more effective in combination. But most interestingly, in the recent times, we have been hearing about plasma therapy that also was used when German flu actually had happened almost a century ago, where they actually take a person who recovered from the infection, his plasma is taken, his blood is taken, they separate the plasma from that, and antibodies that are present in them are injected into the patient suffering from the problem. So antibodies would directly attack the virus. So this again comes under what is called passive immunity that we study under immunity. Okay, so these are the different concepts I wanted to talk to you with respect to coronavirus. Okay, get hold on the concepts, and don't worry too much, because there's so much of information coming, and don't try to understand every aspect of it. That will confuse you further. But with a proper understanding, plan and work on practicing writing this type of information. Okay, so I hope this is useful to you in figuring out what information you need and how should you focus on this. And I'm hoping that that you would prepare well and